So good afternoon, everyone, and thank you again for joining us as the EduSearch Series of ISCB-SC RSG India with EMBL-EBI. In the second episode, we have Dr. Cristian Escobar from PDBe, and the title of this topic is PDBe API Deep Dive. So, as we go. As you know, so basically, first of all, we introduced ourselves as ISCB-SC RSG India. So our motto is your local window to global opportunities. And I just give a brief background about what is RSG India and what's its student council. So basically, RSG India is a part of the International Society of Computation Biology, ISCB, which was founded in US. And under the ISCB, it's an organization who promotes computation biology and bioinformatics all around the globe. And for that, they have around 30 plus countries. They have student council societies which are named as RSG, regional student groups, and RSG India is one among them. So as you see here, that ISCB, then its student council is known as ISCB-SC, and the student council organization named as RSG India, Regional Student Group India. So for just knowledge of you guys, that we have in India, we have 4 nodes. One, as you see, one in Hyderabad, one in Karnataka, one in Kerala, and one in Tamil Nadu. So Tamil Nadu node is recently has been joined us as RSG India. So these are the members that work with RSG India, and these are the core committee members. These are the events we have previously conducted by RSG India in the past 10 to 12 years. So these are the events. So these are some of the photos of our event that we have conducted in the domain of the computation biology. These are some of the events. Yeah. So PDBe-KB. So as our organization is here, so PDBe-KB events are we have today, and these are some of the slides that about PDBe-KB. The PDBe-KB is the Protein Data Bank Europe knowledge base. As you guys heard about it, that whenever we need a protein structure, we will go to the PDB for deriving any structure, and the EMBL-EBI organization has maintained this PDBe web page. So basically, so as for this, as I earlier said, that it is the second episode of the PDBe. The first episode is given by the Joseph. Joseph has told about all the basics things about the PDBe-KB web page, and its lecture is live on our YouTube channel. You can guys, if you want to learn again and you want to see this, you can go our ISCB-SC RSG India YouTube channel, and you can go there and you can check out this video. So as for the second episode, we have Dr. Cristian Escobar. So Akriti will introduce him. Akriti, you can introduce yourself.
Yeah, thank you, Jatin. Am I audible to everyone?
Yes, sir.
Yeah. So thank you, Jatin, and welcome to our speakers and to our audience. So continuing today's episode, it's my absolute pleasure to introduce Dr. Cristian Escobar, and he joined EMBL in 2023 as a database curator, scientific database curator for Protein Data Bank Europe, that is PDBe. His work focuses on the annotation of macromolecular 3-dimensional structures submitted in PDBe, which all of us use very significantly and relevantly for our work. And as we say, data is the new oil. Data curation definitely is the refining process. So he is one of the significant refiners all over the world for the data we are using. In addition to curation, he contributes to development of new features that enhance user abilities and utility of the interface and accessibility for scientific community. And additionally, he is also involved as of today in training activities that showcase PDBe's diverse resources and for exploring the extensive collection that these guys have at PDBe. So Dr. Escobar, he has completed his PhD in biophysics from Florida State University. And during his PhD, he worked on membrane proteins and from Mycobacterium tuberculosis using NMR and spectroscopy. Further, he continued his research as a postdoctoral fellow at Wisconsin Medicine, and he studied RNA G quadruplex structure and protein RNA interactions during his postdoctoral. After that, he's been in curation, and we are very happy to have today's class with him and learn from him. Today's masterclass will walk us through the PDBe-KB's interface, feature, tools, and how these resources can be utilized for bioinformatics research, integrating academia and industry. So over to you, Escobar, and thank you for joining us today, Dr.
Thank you so much for the introduction. So can you see my screen right now?
Yes.
Yes. Okay. Awesome. So today I'm going to talk to you about APIs available at PDBe. As you already know, APIs is everything about gathering data. So as the introduction you listened, data is really important right now. So what I'm going to show you today is how you can gather the data easily in a programmatic way, so you can streamline your analysis process. So this is the list of topics that I will be talking to you right now. So I want to show you what type of data is available at the PDBe, what kind of functional data we add during the different process from annotation and also in our pages. And then I will move on to the what's an API, why you should use your API during analysis, and also the types of API that we have available at PDBe, so they are useful for you. Then we will move on to some examples of these types of APIs. And then I will talk about a little bit about what other resources you can use that we have available for you, so you can learn more about this. And finally, we will go through a small hands-on session where I will share with you some Jupyter notebooks, so you can follow with me in the process of gathering some data and the process of analyzing it. Okay, so what type of data is available at the PDBe? So the protein data bank is an archive of experimentally determined two-dimensional structures, mainly from biological macromolecules. So this corresponds to proteins, nucleic acids, and also the ligands that are bound to these different macromolecules. So PDBe in Europe belongs to the Worldwide Protein Data Bank, which is a consortium of different sites which work together in order to take the data from the scientists, annotate it, and then publish it to the public. So it's for you to analyze later on. So you might be familiar with RCSB, who are in charge of processing entries from the America and also Australia. We have BMRB, who are in charge of analyzing the data from NMR. Here in England, you will find the PDBe and also our partner from the EMBL-EBI, which are in charge of processing electron microscopy data. And then in Japan, we have PDBe, who are in charge of gathering the data from Asia and also now working together with PDBe China. So the final process of gathering this data from scientists, we annotate it, and then we collect it in our archives, and then we make it available to you for analysis. So right now, we are really close to having around 240,000 structures. This is the number up to today, and I guess in a couple of weeks, we will reach that number. We usually process around 300 entries a week. The entries that we receive mainly come from 3 different methods. One of them is X-ray crystallography, solution NMR, electron microscopy. And here in this table, you can see the proportion of each method that contributes to our archives and how the numbers have been changing with time. You can see right now that the number of electron microscopy entries is increasing rapidly, and it has overtaken a solution NMR. We think that in the future, there will be more electron microscopy entries than X-ray crystallography, because of how they can gather the data right now is really efficient and fast. And we get right now even more entries from electron microscopy than X-ray. So, as you know, we collect these three-dimensional structures which correspond to the coordinates of the proteins as determined by the different methods. But those coordinates are not really useful unless we provide certain context. So the data that we provide to you has been annotated, and that information initially comes from depositors themselves, which provide information about how a different structure was collected. For example, the type of method, the sequence that are found in that particular entry, the ligands, and so on. And then we take that data and we process it in our one-depth system, where you can see the diagram right here, which is a kind of linear process. We take these entries, we process it, for example, for ligands in order to standardize the different ligands in an entry. That allow you to compare, for example, the same ligand in one entry with another, because we standardize, for example, atom names, so they can be easily compared. Then we have kind of sequence processing, where we try to map the sequence in an entry to UniProt. And then we try to identify, for example, mutations, expression tags, so you can have that information available. Then in the annotation process, we have another module where we add additional information. For example, we standardize secondary structure, links, definitions, and assembly information. Finally, the entry goes through a validation process where we check the different parameters so we can help you to evaluate if a structure is good or bad in aspects like geometry, how the structure fits well or not with experimental data. So then we provide you with a validation report, which you should take a look into it so you can analyze if the structure is good or not. And finally, we have also part important of the annotation process is communication. So we can communicate with depositors in order to kind of make changes, update the entries, for example, if it has an error. As you can see here, this process is linear, but it's also iterative. So we can go back and make fixes according to what depositors tell us. So this is the data that we are doing annotation, but we also have additional data. And one of our projects that we have currently is called SIFs, which allow us to integrate even more data into an entry, and we provide this information in our entry pages. So in this diagram, you see that there's a close relationship between PDBe and also UniProt, because we annotate our entry and try to map them to UniProt. And sometimes when we are not able to map these entries, UniProt will take a look at those entries that are unmapped and try to find a good map for them. Also, we try to add information about the sequence annotation from Gene Ontology, Interpro, and PFAM to identify, for example, different sequence domains. And also using Escobar and CAF, we try to do an annotation, but in this case from the structural point of view, in order to identify different structural families. So I'm gonna give you just an example of how we present this information to you currently. So this is an example of an entry page where you have, for example, the entry of title, information about the method that was used to gather the structure, macromolecular information here, and structural analysis, and also the ligands that are present in this particular entry. And then the experimental data and validation. So if you look into, for example, the macromolecular section, you can see all the different annotations that we provide to you, which are accessible through each one of these links. And also we provide you with these diagrams, in this case, where you can see how these annotations are mapped to the actual protein sequence. And this could be, for example, secondary structure, validation annotations, and also different binding sites that you can go there and explore. And then now currently we're also providing a ligand information analysis, which is really interesting because now we perform an analysis that will show you which kind of part of the protein is interacting with the ligands. And you can see here in this diagram on the right, all the residues that are interacting with this particular ligand in that entry. And also you can see the three-dimensional view, so you can analyze how well the data fits this particular ligand. So now I can tell you that we are in the process of updating our pages. So also we are collecting feedback. So if you want, you can go to this website, take a look to our new entry pages and provide us feedback. So So the next way that we provide you more information is through the PDBe-KB. So I think you already have a master class regarding how to access data into this database. But I can tell you for the ones that are not familiar with it, that PDBe-KB is a consortium of different data resources that contribute different types of annotation regarding to a protein, and we try to combine all this information related to one single entity or entry, for example. And that's why we call it aggregated views, because we try to gather, for example, all the different structures and small molecules that are related to one particular entry in UniProt. And as an example, I can give you, for example, this for the human acetylcholine. So if you go to the KB pages, you can look for the UniProt ID, and it will take you to this page where you can see a summary of the function of this protein, from where it comes from, and also a view of the best or most representative structure. And below it, you will have all the gathered information for all the structures that are related with the same UniProt ID. In this case, you will find 75 structures. All those structures contain 58 ligands, and so on. So you can use information to gather a much broader view of what broader view of a particular type of protein you are looking for. So this is a kind of contrast with what I showed you before, which is an entry page where you can see the information for one entry at a time, but this one is an aggregated view for all the different entries for the same UniProt ID. So now that you know what kind of data we have for you, I will show you how you can access it using the APIs. So first of all, I want to define what's an API. So an API stands for an application programming interface, which is a mechanism that allows you to integrate two pieces of software so they can communicate between each other. For example, if you have used Python in different kind of libraries, for example, with pandas, for example, pandas is written in C, but when you program, you don't talk to pandas, for example, in C. Instead, you use the API that pandas provide you in order to use Python to talk with pandas and process your data. But in the content that I want to talk to you right now is more related to how you can access information through the internet. This is related to what is called a REST API, where that communication between the programs happens over the internet. So this diagram below shows you how this happens. So usually you are the client, okay? You send a request to the API for certain piece of information. The API processes this request and sends it to our database. Then this database processes the data and sends a response to the API. And then again, the API is the one that sends you the response through internet in the form of data, which could be as a JSON file. So a good way to think about APIs, we can use the analogy of, for example, a restaurant. So imagine that you're hungry and you go to a restaurant in order to get some food. So usually when you go to a restaurant, you don't go directly to a kitchen, isn't it? So what you do, instead, you talk with a waiter, which will take your order, and they take that order to the kitchen in order to be processed, and then it will serve you the food. That happens in the same way. In this case of our API, you don't go directly to our PDBe database to gather data. Instead, we provide you this information through the APIs. So one really important point that it was missing before in the analogy from a restaurant is that when you go to a restaurant, you're looking for a specific piece of food that you want. And in that case, the waiter provides you with a menu, okay? And the menu in the case of an API corresponds to the endpoints, which is a way in order to gather specific pieces of data that you are required. So in that case, in this diagram, for example, if you're looking for ligands in an entry, you need to look for a specific API that will take you and give you that data. And these are just examples of what kind of data we provide to you. This could be in the form of the molecular assembly, ligand information in a particular entry, secondary structure entry, and so on. So I will show you later how you can go and get into the documentation so you can see what kind of data you can get from our APIs. So you can select the appropriate one in order to obtain the information that you want. So why you should use APIs? So in the example that I showed you before, you can go to our websites and take information from one entry at a time. And imagine that you want to analyze 10, 20, or even 100 entries. If you go to our entry page, that process of gathering data is going to be too long, too complicated, and prone to errors because there are so many things that you need to gather. So instead, using APIs, you can do this access programmatically, and you can get this information all at one time, and then use your program of choice in order to process this data. So APIs are really useful for large-scale data analysis. In addition, as I mentioned before, one particular protein might have different structures. So an API can help you to gather all this information from one particular entry, one at a time, all together. So you don't have to do it one at a time. The difference between these different proteins might be the ligands or the condition that was obtained or the different complex that this protein is found. Another thing that is important to why you can use API is repetition of analysis. So imagine that you have created a pipeline for your analysis, and then you can repeat it again with different inputs. So you can repeat that for a different type of protein, for example, or you can repeat that analysis later on as the database grows. So now you might have different entries which can contribute to your data analysis later on. So now, what type of APIs we have available for you? We have 3 types. One of them is querying. This is a Solr-based type of querying system. And this is what help us to, or what runs our PDB searches. So when you go to the PDB entry page, when you perform a search, you're actually using the same APIs I'm going to be showing to you. And this allows you to search for different types of fields. For example, you can look for a certain protein based on the name, organism, experimental methods, and so on. This querying system will help you to gather the entries that you want to process later on, maybe using the other APIs, or if this information is obtained with the search query. Then we have an entry-based API. And this comes from a relational database. This is what we show you in our entry pages. This entry-based API contains specific information from one particular entry at a time. You can gather information about ligands again, different mappings, and all the information that I showed you before in an entry pages. Then we have the aggregated APIs. And this is what we use in order to show you information in the PDBe-KB aggregated pages. So in this case, you can gather information regarding one particular type of protein, and you can get all that information in one go, because this is the same information as the aggregated views. Okay, so if you want to start analyzing data using PDBe APIs, if you want to learn more, I recommend you to go to this API starting page, where there is a description of the APIs available to you. And in this case, you can see the 3 types that I already show you: the Solr-based querying system, the entry-based, and the aggregated APIs. So when you go to the website, you can go to one specific PDBe API and learn about the documentation and the data that is available to you. So first, I'm going to go to an example of the query using programmatic access using the Solr-based querying system. So what you will see in this page is that the data that we provide the search code here provide you a different number of fields that you can use for your search. And these are the same fields that you will find when you perform your search in the entry pages. For example, in this case, the first field over here is, for example, the PDBe ID. This is the information that you need to provide. In this case, the entry ID code in order to perform the search. So you can go ahead and, for example, and look all the different field names, and you can combine it in order to refine your search. So if you go down into that page, you will see this input box, which is interactive. You can input here in the query that you want for your search, and then you can run the code for your example and obtain an immediate response from our API. So to show you the basic structure of a query, so it has three main kind of fields. One in this case where it says Q equal. This corresponds to the kind of the basic structure of the query, where you can see the field name, okay, and the type of data that you're looking. In this case, for example, I'm looking molecule name. In this case, acetylcholinesterase. And then I want to add another term, in this case, for the organism name. So I want to find, for example, all the entries corresponding to Homo sapiens. So this will be in green, your basic search, but also you can have a field list, which is labeled here in red, start with FL, and this corresponds to the terms that you want to obtain from your search. Because when you run a search, you will get a huge number of data. However, with the field list, you can reduce the number of data that you can obtain. In this case, you can specify you only need the PDBe ID, resolution, and the release year, if that's good for you. You can add more if you want, or you can leave it all together in order to receive all the information that we provide. And finally here, WT only shows you what's the format you are getting the information, and usually default is a JSON file. So if you take that query that I show you and you paste it in this query box, and then you run the code, so you will get here below the URL that was used in order to obtain that information. You will find here HTTP status. So 200 means that the query was okay. And then below you see the response in a JSON format. For example, in this case, again, we did a query about the acetylcholinesterase and the organism name, and you can see all the different information that we obtained for that particular query. Okay, so that's the way that you can query our PDBe in order to obtain the information in your search. And once you have the information, for example, for a particular list of PDBe IDs, you can go ahead and go to the entry-based API in order to gather different information that wasn't available with the search in order to find even more information. So when you go to the entry-based API documentation, you can see that we divide the different APIs in different groups, for example, related with the PDBe, compounds, SIFT mappings, nucleic mapping, PISA, validation, and topology. If you go to the search, you will go back to the same query-based search that I just showed you before. So once again, these REST calls are based on different subsets of data, groups of data. And if you go to each one of these tabs here, you will find then examples of how you gather that data that I'm going to show you just right now. For example, if you go to the PDBe API, you can see here that this corresponds to the URL for a specific API. In this case, we're in the PDBe one based on an entry, and also in this case, the particular information they're looking for is regarding to the ligand information for one particular PDBe ID. So below in that box, once again, you have an interactive box where you can input the information that you are looking for. In this case, the PDBe ID, you need to introduce PDBe code as a 4-character string. Then you can press run call in order to run this call and gather the information once again. So in this case, when you run the call, you will get once again the URL that was used for that code, in this case here. You can even take this URL and paste it, for example, in your browser, and you will see that you get the same information that is shown below. In this case, we run the information regarding which ligands are found in this particular entry. And the JSON file here is showing you that the ligand that was found in this case was retinic acid. You can see the CCD code here. On the, for example, more information regarding the weights and so on. Also, if you want to gather more information from different number of entries, instead of doing, you can pass a list of PDB codes here in post data instead of PDBe ID on top, if you want to gather a bit more information from different PDB codes. So now we can move on to the aggregated API. So as I said before, this one is related to gathering data which is related to the, for example, which have been aggregated, for example, by UniProt ID. And this one works similar to the entry-based API. We can see here on the left a list of all possible API calls that you can carry out, and these have been grouped by different topics. For example, in this case, on top here, you can get information about compounds or PDBe, and you can find later on there are even other more groups where you can find. And this one works similarly to what I showed you before. So if, for example, you want to find all the ligands binding related to a particular entry, in this case, we're going to go to the ligand sites, and the information that it provides you is the ligands, the residues that are interacting with a particular ligand in a particular UniProt accession code. Once again, when you go to this particular API call, it will show you the the URL that you need to use in order to access that data. And below, you can see the parameters that you need in order to perform the search. In this case, you need an accession code, which corresponds to UniProt ID. So once you input that accession code into the URL, you can run it and gather the information. Then the documentation will tell you information about the output. So here's a description of all the information that's going to be output during the call. For example, the sequence, and you see the description for this particular entry. And on the right here, you can see an example of in JSON format for the output. So in the case of sequence, you can see here, it corresponds to the sequence of the macromolecule found in that particular entry. Then the length, for example, in this case, 413 residues, so on. And here the important file in this case is data, which contains information about all the ligands that were found interacting with this particular accession code. In the aggregated API documentation, you also can have an interactive box where you can input, in this case, the accession code, and then you can send the call, and you will get the output immediately for you to analyze. And once again, you can take the URL that was output, and you can use it in your web browser to gather the information as well. So do you have any question regarding the APIs at this point? Yeah, if not, I want to move on to other resources where you can get information about our APIs. So we have a kind of seminar web series where you can watch those webinars regarding more specifics of APIs use. Again, you can go to the API start page where you can gather information, documentation for each one of the APIs, and you can get there to the specific site. And also we have a GitHub page with the API training repository where you can go ahead, and we have a bunch of collection of Jupyter notebooks that you can use if you want to learn more about gathering data programmatically using our APIs. The example that I'm going to show you later are examples that were taken from these tutorials. So finally, we're updating also at PDBe our APIs. So I showed you before that we have 3 different kinds of APIs. So right now we're going to be updating in around, I think, 6 months. Is instead of having all these different groups, we're going to put everything together in one single documentation page where you can find all the APIs together grouped by resource. So you no longer need to go to the different 3 websites. So you now go to one and look for all the information that you need. In addition to this kind of changes in the architecture, also there was changes in the framework that manages the API. So now you can have more improved response time. So it's a huge improvement on how you can access the information from our APIs. And in addition, also this new documentation page will have again interactive documentation where you can test your calls before running it in your programs, which is much more helpful for you. So if you want to know more, you can visit this website, which is for now is not the official one. Once we roll out this update, this will become the official website, but you can try it out for now before we roll out these changes. Okay, so now we will move on to a hand session. So if you have any questions, this could be a good time to share your question, while I want to be pasting on the chat a couple of links that you can use in order to access this example that I want to show you right now. Okay, so I just pasted on the chat 2 websites, 2 links to the different examples that I want to go right now. So I'm gonna start with the API search API example. So if you want, you can go ahead and go to the link and follow along to what I'm gonna be doing right now. So can you see my screen right now? I changed it to a website here for the Jupyter Notebooks. So I'm going to start with the search API. So for those who are not familiar with Jupyter Notebooks, this is a way in order to share these examples where you have a combination of blocks where you have text, and also you have programming lines of code that you can run interactively within the notebook. So I'm going to start first. This example, I'm going to show you how to run a search programmatically. So you can start by importing, in this case, installing one of the modules that we need, in this case, SolarCube. To run each one of these programming cells, you can press here the button on the left top corner over here. And you can see that right now it's downloading the module, and it was successful. Okay, and then you can run the next line of code here, where we will import the different modules that we're going to be using, in this case, SolarCube, mainly request, and pandas. So then I'm going to set up 2 main functions. So this function is what I'm going to make the call. That going to help you out to run the call every time. So first of all, the first function is going to kind of create a dictionary that we want to take, and then we want to send in our request. And the main input in this case is the search terms that you're going to be using for your search. In this case, for example, if you remember the previous one, it was about the molecule name, like acetylcholinesterase. We also define the basic URL for our search. This is where we are going to post our search options. Then the second function will take the information about the search dictionary that we defined above and actually create the request. You can see here in this line of code, we're using the request module in order to post our search dictionary to the search URL that we defined on top. And then once we get a response, we're gonna return it as a JSON object. In this case, it will be a dictionary. Then finally, the final function here, run search, is what we all take all the inputs that we want, in this case, the search term and the filters, in order to run this code for you. And it will process the response in order to get directly to the information that we want. So in this case here, you see it's calling the make request function that we created before in order to get a response. And then in the next piece of code, they want to take this response and take some information out of it so you can analyze the output. So then we're gonna run an example. So we're creating the search here. In this case, we want to look for the molecule name for the acetylcholinesterase. So once you define these terms, we pass it to the run search function that we defined before in order to get the information. And then here you can see it's printing what were the search terms. You can see this is a dictionary that we send in our code. It's printing the number of results that we obtain. In this case, we're 10, because that's the number of maximum output that we defined in our previous definition of the function. So in the next one, you can refine your query. You can add more search terms. So before we only had the molecule name. But here I have added the organism name, in this case, Homo sapiens. And also here below in green, you have different examples of other ways you can use the search term for query. For example, you can find things by the EC number, the PFAM name or accession code, Interpro, again, accession codes and names, and UniProt accession codes. You can, if you want, you can run those examples and see what you get out of these different codes. Okay. So we run it. Again, we get 10 results out of it, which is, again, the maximum. And if you want here, you can print the result for the first item in the result that were refined. So if you print it, you will see that we obtain a huge amount of data. This is a really large dictionary with different pieces of data that you can use for your analysis. So that's why it's important that you refine your search using filter terms, because this is sometimes might be too much and there's information that you might not need at all. So that's why in the next piece of code, what we're going to do is to look at the different terms that are found in the dictionary before, and only show you the ones that are important to you. And those are the ones that don't start with Q underscore or T underscore, because those are kind of internal names that we use in the dictionary. And if you run that, you will print all the different useful terms that you can use in your search. Again, it's a huge list of information that you can filter and also use for your search for filtering. So now, for example, in the next piece of code, we can print a specific part of information from our refined results based on the list of information. So we only need, for example, the PDBe ID and the resolution. And once you run it, you obtain these two pieces of data from before based on the different useful terms that were found in the dictionary. So then So you have your list of different useful terms, and now we can create a list of these filter terms. So we only need information about the PDBe ID, resolution, and release year. After we run the search term, we are looking for acetylcholinesterase from Homo sapiens. You can have different lists for filter terms here. I'm showing you a different example. So when you run this code, you can see that instead of getting that huge list of dictionaries with different information that you might not need, now we are only getting information regarding the PDBe ID, release year, the resolution for this particular entry that matches your search terms. In this case, acetylcholinesterase from human. So now you are getting closer to what can be useful for you. So you're not getting that huge list of information. So now you see we have here a list of dictionaries. You can take the information and pass it to loaded spreadsheet. So this function is actually doing that. It's taking that list of results. The output is a data frame which you can use later on for filtering and data processing, which is really common in data science. Okay, so you run the next call, and what we are doing is converting again this list of results that we had before into a pandas dataset, and we are printing only the first top 5 results. And now you can see a really nice kind of a spreadsheet, like where you can have the PDBe ID, release year, resolution, and this can help you out with data processing a lot, because Panda has a lot of functions that can help you out for filtering, for example. So now that you have your nice data frame, you can do things like collecting statistics or printing or plotting some kind of results. Yep. So now, for example, what we're going to do is to analyze, for example, how many entries were published a particular year. So in that case here, this code, what it's doing is grouping the different columns by the PDBe ID. Okay. And we are going to find only the kind of show you the unique values. And in this case, what is showing you here that for each year, you get a number of entries that were published for this particular type of protein. And then you can take that data set, okay, and run it through this function, which will plot the information that you obtain in your column, and you will see this nice graph plot, a bar plot, where you see the number of entries that were released per year. So this is just a simple example of things that you can do. Different data analysis, in this case, just the number of entries per year. Here's another example where you can analyze the resolution, and it shows you what number of entries you have per a certain particular resolution. So yeah, so this is just a couple of one example of what you can do with search. So the main idea is that you can use the search query in order to find particular entry you want, filter by the information you need, and then take that information, process it, or even use it to run another queries or calls for the other type of APIs. Okay. So do you have any questions regarding this? Yeah, if you don't have questions, you can post it later on, so we can have a talk to the different topics. So I will move on to the next one, which is instead of a search, I'm gonna go through an example of the aggregated API. The way that this works is quite similar to the entry-based API. Okay. This example is going to be a little bit more complicated. I mean, there's going to be a lot more data processing, but it's just an example to show you how powerful is the API search. So you can do all types of data analysis. So I hope that this one is useful for you. Okay, so in this case, what we're going to look is into a ligand interaction. So we're going to make a call to a specific API where we will gather information about which residues are interacting with a particular ligand for a particular accession code, UniProt accession code. So in this example, we're going to look again to the human acetylcholinesterase, and the UniProt accession code is this one. We're going to use that throughout our calls. So first, once again, run the first cell in order to import the module that you will need. So it's going to take a couple of seconds to run. Okay. And then here I'm defining two functions. One of them is going to take the URL. I'm going to send the call to our API. And it's going to take the response and give it to you as a JSON file. So in this case, the calls are much simple, and you just need the URL that I showed you before in the other examples for the different particular API that you want to access. So in this case, the base URL you can see here is this one. We're going to then define it a little bit more. So we're going to use the graph API, which is related with aggregated API, and then which is going to be by UniProt ID. And then the next function is actually going to take the accession code that you want and make the call for a particular URL. So now here we're expanding the URL, and we're going to be looking to ligand sites. So, and then we are going to add the accession code, in this case from UniProt. We're going to run the call using our get URL information, and then you will get information as a JSON file. Then you will take the information from that JSON file, and we're going to kind of filter it into certain parts of the data. So because there's some information that you get that you, in this case, we don't need. So this function is going to go there and take it and use it. But something important that we're defining in this function here is we're going to calculate what we call an interaction ratio. So remember that we're looking at two residues that are interacting with a particular ligand. And what this part of the code does is to count how many times a particular amino acid in an entry is interacting with that ligand based on the total number of entries where that ligand was found. For example, if you have ADP and you have an alanine that's interacting, you have in 3 structures, and it's interacting 3 times, then the interaction ratio is going to be 1, because you have 3 structures and you see that the same alanine interacting with the same residue 3 times. Okay, so that means that the ratio is below 1. That means that that particular residue is not interacting with that ligand all the time. Okay, so that means that ligands that are interacting with an interaction ratio of 1, those might be more important than the ones that have a lower number. Okay, so we're gonna run the call using our accession code here for the human acetylcholinesterase. Once again, you get a huge amount of information, but mainly what you get is correspond to information regarding a particular ligand. For example, in this case, you can see here the name of the ligand, the CCA-DAD for that ligand, and then the residues that are interacting with that ligand in that particular entry. For example, in this case, you see that residue 117 is interacting with this ligand. Okay. So as you saw, it's a huge amount of data, and the next functions, what we're going to do is to simply reprocess it and remove, expand the data that is in there, because some of the columns might have information as a dictionary sets. So what it's doing here is expanding that information into different columns. So in general, what it's doing, this function is to take the data set, which is a list of dictionaries, and then creating a data frame, in this case, using the function explode in order to doing what I just mentioned to it. So, and also that is done in combination with the next part of the code, which is normalize, where again takes those dictionaries that are in a column and creating new columns based on those dictionaries. Okay, so you can go ahead and later on look at this piece of code and go and see what is actually happening in each one of those lines. So what at the end, what you will have after the data frame is clean is a new data frame with all the information coming from the code. So each row corresponds to one amino acid that's interacting with a particular ligand. In this case, for example, tryptophan 117 is interacting with this particular ligand in this entry. Okay. And then you have additional information here and similar information to the end of the data frame. So then what you can do is to take this data frame and do some additional processing in order to filter the data and gather the information that you want. This is what we're going to be doing later. So for example, here in this cell, what we can filter this data frame by the interaction ratio, and we only want to find which residues, which ligands are interacting with residues with an interaction ratio of 1. And then we will filter and only get the unique values. So what we are printing here is a list of all the different ligands that are interacting consistently with the protein. Then you can get additional information, in this case, per residue. So we can count, in this case, how many times one particular residue is observed interacting with a particular with a ligand. This is what it's actually doing. So it's taking our data frame and grouping by the residue number and counting how many times that residue is observed performing an interaction. This is the final data frame that you're observing. For example, in this case, residue 44 is only observed interacting 1 time. And then, for example, 47 is observed interacting 11 times. So with this information, you can make some basic statistics. The next piece of code is actually taking this same data frame, getting the average value of the number of interactions. So it's looking at this count and making the average value, and also getting the standard deviation. So once you print this data, you can see that in average, one residue is interacting is observed interacting 11 times with a standard deviation of 16. Okay. So now that you have those basic statistics, you can do more interesting stuff. Like, for example, in this case, we're going to plot which residue in this protein are interacting more than others. So, for example, here in this part, on this part of the code, we're going to filter by the number of counts, which are less than two standard deviations, and we're going to make we're going to show those actual residues in blue. And then in the next part of the code, we're going to filter by which residues are interacting a number of times larger than 2 standard deviations. So this is going to be the more significant residues. And we're going to plot these ones in red. So once you plot it, you can see it like this diagram. So you have a diagram with a residue number on the number of times that residue was observed interacting with a residue. So this line corresponds to time the standard deviation, and anything below that is not going to be significant, and anything above is going to be significant. So this already is showing you important information if you are thinking about, for example, modifying your protein you wanna kind of change how it interacts with a particular ligand, because you can see which ones are the important ones. Okay, so so now that you know which residues sorry, are actually interacting significantly with others, you can use that information to find which are the ligands that are interacting more significantly. Okay. So in the next piece, when you, once you run it, it's showing you a list of which residues are over 2 times the standard deviation, and this column over here is showing you the list of those residues. Okay, and then in the next one, we want to define which are the ligands that are kind of interacting the most with these particular residues that are important. Okay, so for that, what we're going to do is to look at our data frame. We're going to look at the residue number, and I'm going to check if that residue is in our previous list of residues that interact significantly with a ligand. And then we're going to take those, the list of ligands, only get the ones that are unique. And you can see this corresponds to the list of ligands that interact more significantly with our protein. So then, so you know the list of the significant ligands, and you can also get which ligands are not significant. For that, you can simply get a list of all the ligands in your entries in this piece of code, and then here, using this list comprehension, you can find the ones that are not found in this list of significant interacting ligands. So these are the ones that are not significant at all, which makes sense. For example, you have magnesium, which is just a salt, so of course it's not acting significantly. And then using these pieces of information now, you can filter and gather information about which ligands are interacting, and also the residue number and the interaction ratio. And that's why this piece of code, what it's actually doing is grouping again by the residue number, the ligands that are interacting, showing you here the average interaction ratio. And this will help us to perform some additional analysis that you will see in the next figure. So what we're going to do is we're going to take the information in this data frame, and we're going to plot, in this case, a scatter plot. In this case, what we're going to do is to filter, okay, by ligands, which have an accession code. In this case, this corresponds to the ligands that are not kind of interesting, okay? And we're going to plot those ones in blue. The size of the dot is going to be in proportion to the interaction ratio. Okay. And then in the next part, we're going to do the similar, but in the case, we're going to filter by the ligands that are more interesting. We're going to show those ones in red. And what you're going to see when you plot this is correspond to this nice plot where you have the residue number. Okay. And then on the left axis here, you will see each particular ligand that was found on the different entries. So remember that the blue ones are the ones that are not interacting significantly, and the red are the ones that interact more significantly. And here you can see each dot corresponds to one particular area. So what you're seeing here corresponds to which residues are interacting most significantly with this ligand. And you can see these lines that correspond to residues that constantly interact with different ligands. This can give you a first glance which residues are actually important for ligand interaction in this protein. Okay, so again, this is one of the an example, a little more complicated one, but just to show you how powerful is the API's information that we provide, that you can use process in different ways. In this particular case, using information of which residues interact constantly in a protein with different ligands. You can go ahead, for example, if you're trying to design a particular drug binding molecule, you can use this information in order to see what information, what might be relevant or not. Other kind of thing that you can do, for example, is to analyze all these ligands and see, for example, if there is some relationship between them. Do they share, for example, a similar backbone? Can you use that information for drug design, for example, and so on. So, yeah. So yes, so this is what I wanted to share with you. So if you have any question, I will be glad to answer anything that you might want. So if something wasn't clear, yeah, just let me know.
Yeah, thank you, Dr. Cristian. I think it was very informative talk, and so today we have seen the true potential of the PDBe APIs. So this is just like ChatGPT for us. So those who are going to work on the structures seriously, so you give the query and we're getting whatever we want, and we can filter everything. So it's a great demonstration as well. Yeah. And
Yeah, so
we have, please.
So again, yeah, you just can use this in order to gather information you might need for your AI, for example, training models. And of course, you need to clean it and prepare it accordingly. But yeah, the information is there. This information has already been prepared for you to use. So yeah, just need to go ahead and use it for your purposes. So.
So we have few questions here. As you just answered the question, the same question, how can we retrieve these structural features and annotations that are suitable, so that can be readily used for the AI or ML models?
Yeah. So as I said, depending on information that you need for your training, AI models, all this information is available to you. You need to just find it according to the documentation. Of course, it needs to be pre-processed for if you want to use it for AI. It might depend on what you want to do. For example, working on some kind of name entity recognition for certain data that is available. You need to pre-process it, gather it, pre-process it, and then use it for your model. So
we have another question. So for this PDBe aggregated APIs, can we use these APIs on our own data, own generated data? For example, molecular docking, and so we generated the data through docking, and we have protein ligand interaction data. So can we use these APIs to check all these interactions and everything?
are you saying to run the analysis on other data?
Yeah, excluding the data from the PDB. So
the
our
thing
own
is that
generated data. Yeah.
The thing is that the data has been pre-processed for you. So with the API, we're making it accessible. So we cannot process your own data with it. That's a kind of distinction. But for example, in the case of this ligand interaction that I show you, we use some pipelines that are available for you. So in that case, you might need to go, for example, go to the GitHub repository, download the piece of software that you need, and then repeat the analysis for your own entry. So.
Okay. So audience, if you have any questions, you can type in the chat. In the meanwhile, if anyone wants to ask the question, you can.
Yeah, Govind, may I ask?
Yes, go ahead.
Yeah. Thank you, Dr. Cristian, for the informative session. I wanted to ask that, just out of curiosity, do you have similar APIs for updating the database at PDBe? Like, how frequently do you update the already existing queries, already existing records at PDBe? And does similar kind of system as the search APIs you just demonstrated exist for updating those as well?
Yeah, so we at the PDBe, we update our repository every week. So every week we release new entries. During that release process, we recalculate all the information that you're seeing for those new entries. So you can go ahead and run your pipelines. For example, the code that you want anytime you want. So it's going to be updated. So usually we release on Wednesdays. So after Wednesday, it's going to be updated. So you can check your new calls after Wednesdays.
So the updating is also automated, that means.
Yeah, so everything is automated during the release process. So yeah.
So audience, if you have any questions, you can ask the question directly, or you can. Okay. So we have one question here. So as we have mentioned about API, but do we have modules or tools to extract information from the API output files?
Okay, so yeah, so as I show you, we provide, when you run the call, what you get is a JSON file, and then you need to create your program in, like I show you, is in Python, for example, in order to extract the data out of it. So yeah, that's the process. So remember that this is already pre-processed for you. You get the JSON file, and then from the JSON file, you extract the data that you need for whatever purpose your program is actually doing. So.
Okay, thank you, Dr. Cristian.
Yeah.
So I think this is the personal question for you. So what are the current challenges in curating structural data for membrane proteins, and especially those from the pathogen bacteria like Mycobacterium tuberculosis?
Yeah, that's a good question. So, I mean, right now we don't make a distinction of memory protein from other kind of proteins. So we apply in the same, for example, validation tools to all the proteins in the same fashion. So that's a good question. It would be great to have different validation metrics for memory proteins, because the environment, of course, affects what the structure of a protein. And yeah, we have seen reports of, for example, having ligands where it shouldn't be, for example, something detergent inserted in the membrane, which might affect the structure in that particular region. So unfortunately, yeah, we don't have validation tools that differentiate or that kind of which are particular for that. So all of that might depend on the analysis of the person who is looking at the data. So that could be one big challenge. So.
Nikita, is your question is answered, like, or you can ask the question maybe?
Okay, yeah, I can see a question about the format predefined.
Yeah.
So yes, so you can go ahead and as I show you in the documentation, and there are examples there of the output data. So you can go ahead and look at it. Each call might give you different data, so the structure is going to be different for each response that you get. So, but everything is documented, so you can go ahead and look at it and see the output that we provide. So.
Thank you, Dr. Cristian, for the excellent demonstration.
Yeah, you're welcome.
Yeah.
You're welcome.
Yeah, I think these APIs will be very useful for everyone who is going to work on the PDB and especially the protein structures. And yeah, so we can make it public and everyone will use it. Jana, can you
Yes.
take the lead? Yeah.
Hello. Yes, sir. So thank you once again, Dr. Cristian, for such clear and elaborative, insightful masterclass. A big thanks to every one of you joined here today. I would also like to extend my great heartfelt gratitude to the teams of EMBL-EBI, PDBe-KB, and of course our amazing RSG India, our amazing RSG India volunteers. So that's a wrap for episode 2 of EduSearch series. But stay connected. We have many more series many more episodes lined up as which are designed for to connect knowledge with the real world applications in the field of computational biology. We'll be sharing the link. I'll be sharing the link in the chat box right here. Just a second. So I request you all to please fill this feedback form and do follow us on our RSG India on channels, social media channels. You can also visit ISCB websites and also our RSG India website for more opportunities and updates.